Good morning, everyone. Now we are ready to start the session. Good morning. Is it audible now?
Yeah, it's audible.
Okay, thank you. First, I wish to confirm the presence. Dr. Bahiruddin, are you here?
Yes, I'm here. Yeah.
Okay, thank you. Yes, doctor, it's okay. So then participants, please confirm your presence. Miss Subhashini, are you here?
Yes.
Okay. Mr. Harichandra?
Yes, madam.
Okay. Miss Aradhana?
Yes, ma'am.
Jayasinghe? Okay, thank you. First, I wish to introduce our panel members. I am Dr. Niranjana. I am the chair of this panel. I wish to introduce Dr. Bahiruddin. Bahiruddin, could you please tell about you?
Yeah, working as a department of agricultural biology. The IIC post pathogen resistant breeding cultural biology. So I did my PhD in University of Sydney. Senior lecturer.
Okay, thank you, Dr. Bahiruddin. Hello, Dr. Prasanna. Could you please introduce yourself?
So good morning, everybody. So I am Dr. Prasanna, senior lecturer attached to the Department of Agricultural Biology, Faculty of Agriculture, Eastern University. So I completed my PhD degree at the University of Queensland last year. So I received my duties at the faculty last year, 2022.
Okay, thank you very much. Participants, I hope all of you are ready now. So I kindly call first presenter, Ms. R. Subhashini, to present your research title on investigation of microbial quantity and diversity in processed cinnamon kept under different post-harvest treatments. Subhashini, now you can start your presentation. Start your presentation.
From Uvula University of Sri Lanka. The research was conducted at National Cinnamon Research and Training Center. As you can see on this screen, my research topic is investigation of microbial quantity and diversity in processed cinnamon kept under different post-harvest treatments. This is the content. Let me start by saying about the background of my research. Ceylon cinnamon or true cinnamon is one of the major spice crops export from Sri Lanka, which belongs to the Lauraceae family. It's a large evergreen tropical tree that grows to height about 10 to 14 meters in their natural habitat. The dried inner bark of this stem is used to make cinnamon coir. Sri Lanka exported around 18,000 metric tons of cinnamon and worth of 38 million rupees in 2020. Sri Lanka supplies more than 90% of true cinnamon to the world market. Let's move to the problem statement. Microbial infection of processed cinnamon leads to the quality defect and considerable post-harvest losses. Different storage conditions accommodate various fungal species and may create fungal colonies on processed and stored cinnamon. This may violate the quality standards of processed cinnamon, thus the product be seen of low quality. Moisture content of cinnamon, temperature and relative humidity of the storage are most important variables in managing the microbial growth, so that proper storage and management protocol could be defined to overcome this quality defect. Next to the justification statement. quality of the processed and stored cinnamon has become a huge barrier in the Sri Lankan cinnamon industry. It causes severe issues via rejections of cinnamon consignment and damaging the international reputation of Ceylon cinnamon. Although isolated experiments and reports exist on the subject, the research gap has not yet been bridged well to record the levels of environmental variables of storage cinnamon against the diversity and intensity of microbial contaminants. Let's move to the objectives. The general objective is to study the microbial quantity and diversity in processed cinnamon stored under different post-harvest treatments with the time and moisture content. Also, there are 3 specific objectives as mentioned here. This flowchart shows the method of my research. First step was the sample collection. For the study of moisture decline of cinnamon, 500 grams of freshly processed cinnamon quills were obtained from the cinnamon training center. Then for the 6th treatment of the main study, 7.2 kilograms of cinnamon quills were obtained from the cinnamon storeroom, which were maintained 14 days from the drying room. Second step was to determine the moisture decline in processed cinnamon. For the explain clearly, I like to show this picture. This is the primary study, and it was done to identify the most reliable instrument for recording the moisture level of processed cinnamon. For that, freshly processed cinnamon quills were obtained and moisture content of the sample was periodically measured using moisture meter and moisture analyzer. Average moisture content was recorded twice a day and moisture decline was drafted up to a week period. Next step was the studying of fungal contamination. There were some steps to isolate the emerging fungi in cinnamon quills. So, culture were prepared via single spore isolation technique. Then the colony, mycelial and spore morphology of the isolates were identified, and slide culture technique also was followed. Fourth step was the applying different post-harvest treatments. Samples were tested in triplicates and 200 grams of cinnamon quills were used for each treatment. Then 3,600 grams of processed cinnamon quills were treated with sulfur dioxide using 45 grams of sulfur for 14 kilograms of cinnamon quills in a closed chamber for 12 hours. Cinnamon quills were kept in a regular storehouse for the control treatment. And other 5 treatments were applied according to this table. At the fortnight interval, 50 grams of cinnamon quills were taken out from each replicates and tested for moisture content, colony forming unit, and fungal diversity. Next, move to the data collection part. Colony forming units and fungi. Colony forming unit of fungi and bacteria were recorded after 3-day incubation period. The colony forming unit for all the treatments were calculated using this formula. The Simpson diversity index was used to determine the fungal diversity of stored cinnamon quills. Number of different fungal species such as Aspergillus, Penicillium, Rhizopus were recorded that had grown on petri dishes after incubation for 3 days. Let's move to the result and discussion. The different types of commonly emerging fungi on cinnamon quills were identified using their morphological and cultural characteristics. There were specific variation in the colonies and colonies as macromorphological features. This figure shows the morphological changes of colony surface of Rhizopus, Penicillium, Aspergillus, and Trichodema-like fungus for species with time. This table shows the colony surface and colony rivers of the different fungal species. Let's move to the specific microscopic characters of the particular species of fungi that was observed using dissecting microscope. Here, this graph shows graphs accommodate to identify the most reliable instrument to get moisture reading of processed cinnamon. The moisture content of cinnamon quiz observed significant variation among the moisture meter and moisture analyzer. Although most of the farmers use moisture meter to obtain moisture level in stored cinnamon, according to our result, moisture analyzer showed higher accuracy and it can be recommended over the moisture meter. This graph shows how the moisture was declined with the time in freshly processed cinnamon. For this, data were collected up to a week period. The moisture content of the cinnamon has decreased within 3 days, as well as the moisture reduction in processed cinnamon is faster when cinnamon quills are stored in control conditions compared to the storing normal cinnamon storage. This graph shows the number of fungal colonies were grown on processed cinnamon quills stored under 6 treatment with the time. The lowest number of colony-forming units were observed in treatment 6 and treatment 4, respectively. When cinnamon quills were treated with sulfur dioxide, the fungal contamination was reduced until 6 weeks and increased after a 6 weeks period. Storing cinnamon quills under control condition with sulfur dioxide fumigation reduced the colony-forming units significantly than the control treatment. Cinnamon quills stored in sealed polythene bag and stored in the storehouse increased the fungal contamination. The most favorable condition was the treatment 6, which was observed lowest number of colony-forming units. Let's move to the bacterial CFU graph. According to the graph, the lowest number of bacterial colony-forming units were observed in sulfur dioxide fumigated cinnamon stored in sealed polythene bags. All the other 4 treatments were performed well, as the bacterial colonies observed in constant level, except the treatment 6. Bacterial colony-forming units of cinnamon quills stored in the storehouse was higher than the other treatment, as well as sulfur dioxide fumigation and storing in sealed polythene bags was the best treatment for storing cinnamon quills against the infection bacteria. This graph shows the different fungal species such as Aspergillus, Penicillium, and Rhizopus growing cinnamon quills with time. The lowest fungal diversity was observed in treatment 6 and treatment 3, respectively. Here, fungal diversity was gradually decreased when cinnamon quills were treated with sulfur dioxide. Also, fungal diversity was higher in the cinnamon quills stored in the storehouse and stored in control conditions. This graph shows the moisture content of stored cinnamon quills measured using moisture analyzer with time. As you can see here, cinnamon which was stored under control conditions showed the lowest moisture content within 2 months period than other treatments. Cinnamon which was treated with sulfur dioxide showed the comparatively less moisture content within 6 weeks period. Let's move to the conclusions. According to the macro and microscopic observation, Rhizopus, Penicillium, and Aspergillus species were identified as the most common fungal contaminants of stored cinnamon quills. The sulfur dioxide fumigated cinnamon quills stored in both control condition and sealed polythene bags remained with significantly low microbial contamination throughout the observation period compared to the control treatment. Significantly reduced moisture level was observed when cinnamon is stored under control conditions. And the sulfur dioxide fumigated cinnamon quills stored in control conditions showed significantly low fungal diversity, bacterial and fungal conforming units throughout the observation period than the other treatment. Treating processed cinnamon with sulfur dioxide and maintaining them under modified environmental conditions with reduced moisture and relative humidity can ensure an extended shelf life with reduced microbial contamination. Finally, as the suggestions, it is favorable to extend of the study time period to get better results. Also, this study should be repeated under different environmental conditions than using 1 or 2 temperature conditions. As well as these findings should be introduced to the respective points of the cinnamon industry to follow those things. Finally, molecular identification will confirm the different fungal species that have been identified morphologically. These are the references. That's all about my presentation. Thank you very much.
Okay, thank you, Madhuvanti. Now the time open for discussion.
Thank you, Madhuvanti. Is my voice is audible?
Yes.
Yeah, so I have to clarify a few things regarding your experiment. The first thing is, so once you are testing this different experiment and fumigation and other things, did you check that any other qualities of the cinnamon declined or improved something?
Sorry, sir. Can you ask again?
Sorry?
Can you ask again?
Yeah. I'm asking directly that did you check any quality changes happening or happened during the fumigation process or other experimental trials you set?
Yes, cinnamon colors.
Yeah, cinnamon color and the taste and aroma. Did you check it?
Yes. No, sir. For the experiment, I not check.
And also, did you check the residual effect of that your treatment?
No.
Because in detail study, when you are doing again, you please consider this.
Okay.
Okay, thank you. Thank you.
Thank you.
Madhuvanti,
Yes.
so you measured the fungal diversity, no?
Yes, sir.
Yes, what is the unit for that one? So what parameter actually the fungal diversity mean? So which index you measured for that one?
There is no any parameters, units for the fungal diversity, but there is a equation, and I use it for that fungal diversity index for the experiment.
Okay.
Hello, Madhuvanti.
Yes, madam.
Yeah, is the your finding says that the sulfur dioxide fumigated and packed in the polythene, that is the best treatment, isn't it?
Yes, madam.
The sulfur dioxide no any harmful effect to the human? Have you find any harmful effect by the sulfur dioxide?
Yes, madam. Sulfur dioxide is widely used food additive, and when I use the sulfur dioxide fume, I think there's no any if may damageable effect on the human.
You didn't get any effect, no, by this usage of sulfur dioxide. But for some
Yes, ma'am.
people, it will cause some respiratory problems, isn't it? Huh?
Yes, madam. And
Okay.
European countries, they not recommended sulfur dioxide,
Yeah.
but
Yes.
yes.
Yeah, that's what. So then you should find some other alternative as well. Huh? In your that suggestion, you should mention. Huh?
Yeah, yeah, madam.
Okay, right. Thank you,
Thank,
Madhumati.
thank you, madam.
Okay. Are there any more questions? Okay. Now I kindly call upon second presenter, Mr. Harichandra. Please present your research on control of Fusarium oxysporum causing Panama wilt in banana using plant extracts. Please share your slides and do the presentation.
Okay, madam. Can you see screen, madam?
Yes, it's clear. You can do the presentation.
Have a nice day, everyone. I am basically Harish Chandra, Faculty of Technology, University of Jaffna. My research topic is control of Fusarium oxysporum causing Panama wilt in banana using plant extract. Introduction. Banana is one of the best marketed food in the global market, and it is a nutrient-rich food crop. Banana is cultivated on a large scale in more than 10 million hectares in more than 120 countries. The annual banana harvest is about 8 billion metric tons. Panama diseases of bananas is also known as banana fusarium wilt. The causal organism of banana disease is the soil-dwelling fungus said Fusarium oxysporum. There are many varieties of bananas that can be affected by these diseases. The disease is also more likely to spread in the tropicals. Banana plants affected by Panama diseases showing in the picture. And Fusarium oxysporum fungi sample and objectives to control Panama disease of bananas using green manure to control the growth of Fusarium oxysporum by adding green manure to growing media. Problem statement. To date, no chemical disease control method has been found to control Panama disease in banana. Many banana plantations in Sri Lanka are affected by Panama disease. Fusarium oxysporum clematospora survive in the soil for a longer period without host plant. Therefore, difficult to control Panama disease. Justification statement. An eco-friendly Fusarium oxysporum control system must be found to overcome the current situation. Therefore, the study will examine the green manure leaves recommended to inhibit Fusarium oxysporum. The focus here is on preventing the spread of the Fusarium oxysporum fungus that cause Panama disease in banana using herbal extract from green manure plant. Materials and methodology. Chemicals, instrument, and methodology. Experimental design is complete randomized design. Data analyze method is data were analyzed by using SAS computer software. Firstly, PDA media preparation. Firstly, potato electrode agar convert taken and measure PDA was measured, and that PDA was taken in blast and water 2 milliliter added, 2 liters added, kept in a house plate and prepared for the autoclave and autoclave. And in vitro culture, collection of samples, Panama diseases infected banana stem samples were collected from Gunungkula Palace. Disinfected laminar flow cabinet. Laminar flow cabinet was sterilized using UV rays and alcohol. Then it was turned on and left on for about 20 minutes. Isolation causal organism. Firstly, find the Panama stem samples were collected and the samples were isolated. An isolated Fusarium oxyvorum sample in this picture. Screening of different plant extract for the use of green membrane. Firstly, find the leaves sample and measure the 250 grams of leaf sample and cut into the small pieces and grind the leaves and add the water. Add the water and grind. Then leaves extract get and added agar and glucose. Then keep in on the host plate and autoclave. That sample after that for the petri dishes and Fusarium oxyvorum fungi sample inoculated in this petri dishes. Data collection. Fusarium oxyvorum colony diameter was measured after calculating using below formula. Reduction in growth percentage 100 multiply diameter C minus diameter F divided by diameter C. Diameter C mean growth zone diameter mean of the control plate and diameter F mean growth zone diameter mean of the sample plate. And Fusarium oxyvorum inoculate medium preparing media and banana tuber samples find and cut into the small pieces and add the glucose and mix in well. Then this media filled banana tube filled in the polythene bags and autoclaved. Inoculate Fusarium oxyvorum and then picture showing Fusarium oxyvorum inoculated banana tuber media samples. In vivo culture, selection of plants, tissue culture banana plants about 3 months old without infection was selected. And media preparation, topsoil sand compost 211 red get and mix well. Then Fusarium oxyvorum inoculum added and mix well. Application of Fusarium oxyvorum inoculum media to a banana pots. Firstly, take the 3 months old banana plants, put plant inside the polythene pot and inoculum media some amount get and plant with inoculum. Applying plant extract, plant leaves sample you taken measure leaves samples and add the water and grind it and extract get take. Then added this extract in the plant. Then after applying the plant extract, caring for banana plants, the banana plants were kept in a net house. The plants were watered about 1 hour to days. Result and discussion in vitro culture. Effect of plant extract on the percentage reduction in growth of Fusarium oxyvorum after 2-day interval. Clove and cinnamon successfully inhibit the clove, cinnamon, nutmeg successfully inhibit the Fusarium growth rate. Neem, Glycidia, Malabar like that growth in the extract growth the Fusarium oxyvorum and Moringa castor plant like that enhance the Fusarium oxyvorum growth rate. And 4-day interval, clove and cinnamon successfully inhibited Fusarium oxyvorum. Glycidia, nutmeg grow in neem, moringa, castor plant like that plant extract enhance the Fusarium oxyvorum growth rate. Then 6-day interval, clove and cinnamon successfully inhibit Fusarium oxyvorum growth rate. Glycidia, nutmeg grow in neem, moringa, malabar nut castor plant like that plant enhance the Fusarium oxyforum growth rate. 8-day interval, clover and cinnamon successfully inhibit, but neem, Glycidia, nutmeg like that plant grow in. The effect of leaf extract on the growth of Fusarium oxyforum, the appearance of the second day, the appearance of the fourth day, the appearance of the sixth day, the appearance of the 8th day. In vivo culture, the plants that were good on the first day has turned yellow and withered after about a month. First day of the treatment and after a month later, the part of the system near the root board of the banana plant was rodden. This picture has rodden banana stems. The root system of almost all the banana plants were good condition. The tubers of many banana plants were rodden, but the tubers of the banana plants where the clove extract were applied did not rot. Glycidia first picture and clove second picture did not rodden. In fact, banana plants were susceptible to bacterial rot caused by rainwater due to heavy rainfall of those days. Conclusion: according to the in vitro culture, cloud and cinnamon were the best Fusarium oxyforum inhibited. Inhibition percentage is 100%. In fact, inhibited neem, moringa, malabar castor plant, Rapetia and Sosa stimulated Fusarium oxyforum mycelium growth. In vitro culture, in vivo culture, the bacterial rot of banana plants treated with clove extract was controlled. Recommendations according to in vitro culture result can be used as treatment for banana infected banana plants in the province cinnamon field. In the future, by developing this agreement concept, this method can be tried on the plants as well. Acknowledgement, reference, and thank you very much.
Okay, thank you, Haris Chandra. Now the title is open for discussion.
The Harichandra,
Yes, sir.
so I wanted to ask a few clarifications. So when you apply this botanical extracts to the experiment,
Yes,
so how
sir.
did you do that?
Firstly, finds banana Fusarium oxyforum affected banana stem parts.
No, I am asking just how you applied your extracts to the experiment.
Firstly, find the leaves, cloves, cinnamon like that leaves, and measure the 250 leaves grams and grind in and add 2 2 liter of water and mix
No,
them.
I am not asking your extraction process.
Yes,
So
sir.
how did you do how did you apply the extraction to the treatments?
Yes, firstly, banana plants 3 months like that all banana plants take and affected the inoculum sorry moxyforum inoculum we make and add in the this plant and affected this Fusarium moxyforum fungi in these plants. Then we apply the plant extract. Then
So how
we
did you apply? That's what I asked. How did you apply?
plant extract 75 milliliter like that plants extract get and one plant cinnamon. We select the best plant extract, cinnamon,
No,
and
yeah. How did you decide to apply 75 ml?
that talk with my supervisor, and she.
Okay, okay. Then fine. It's okay. It's okay.
Okay, sir.
Okay, Harishchandra,
Yes, madam.
yeah, could you please take the slide you put previous to the conclusion? Go to the conclusion slide. Yeah, previous one.
Okay.
No, no, no. Previous to this one, that table you put it. No, no. Table you put.
Madam.
Yes. So what is the experiment you have done here? Banana plants.
Banana, some plant, madam. Rodent, madam. Then we get the result rainfall these days.
Yeah, what is the correlation? I'm asking why you have done. You have done that extract, no plant extracts, effect of the plant extracts on the fuse area. Now, suddenly you put this slide, bacterial rot and rainwater. Why you
Madam,
have? Yeah.
we doing this experiment these days heavy rainfall, madam, because some plant uh, uh, we couldn't get accuracy result, madam, because this plant was rodent. Then we had the reason, madam, this for this.
Okay, but fusarium mean that is the fungal disease, no?
Yes, madam.
But this is the bacterial rot.
Yes, madam. Fungal disease. These plants because heavy rain because heavy rain this affected these plants bacterial disease, madam.
Okay, anyhow, this is not clear, but anyhow, you check it. Huh?
One question.
Yeah. Who was your supervisor over there?
Supervisor Alakon, madam. Sir?
In Jaffna University
Yes, sir, yes, sir.
from Biosystem Department.
Yes, technology biosystem department, sir.
Okay.
So one more question, Harichandra, just I am asking, so you mentioned some neem including some extracts promoted or stimulated fusarium growth. So what may be the reason for the literatures, particularly the neem controls reduces the germination of fusarium mycelium growth. So here, what may be the reason to stimulate the growth of fusarium by neem extract and some other extracts? What may be the reason?
Sorry, sir. Not clear that question, sir. Sorry, sir. Can you ask again, sir?
Just directly I'm asking. So you mentioned the neem extract stimulated to the enhance the growth of mycelial growth of fusarium.
Neem extract.
Yeah, you mentioned the conclusion. They stimulated the growth of fusarium. Neem
Yes, sir.
extract. So what may be the reason? Normally, the neem extracts retards, so the reduces the growth.
Neem extract, sir. Neem extract growth wells Fusarium oxyborum fungus growth wells in the neem extract.
So do you have any idea what may be the reason for that one? No. Okay.
Okay, Harishchandra, only one more suggestion. But you have selected clove and cinnamon spraying as a botanical pesticide. Those are the best one, no? But anyhow, I can't believe that how the farmers can bear the expenses of these. Actually, clove and cinnamon are very expensive spices, isn't it?
Yes,
It's better. Yeah, it is better. You have to check that cost benefit ratio as well. Huh?
okay, madam.
Okay, okay. Thank you very any more questions?
No, madam.
Okay, thank you, Harishchandra.
Thank you very much, madam.
Then the third presenter, Miss Aradhana, please do your presentation on study on seed setting performance of selected cluster onion cultivars under vernalization and non-vernalization conditions. Please do it. Yeah, it's okay. You can present.
Good afternoon all. Good afternoon all. I am Aradhana Manivarnan from Faculty of Agriculture, University of Jaffna. Today I am going to present my undergraduate research study on seed setting performance of selected cluster onion cultivars under vernalization and non-vernalization condition. Let's move to the slides. This is my acknowledgement. Thank all of the staff members of Faculty of Agriculture, University of Jaffna, and I wish to thank all of the staff members and workers of Plant Breeding Division, RARDC Klinochi. And I would like to thank those who are providing support during this study to make my research worthwhile. First one, introduction. Allium cepa is the onion. The onion is mostly cross-pollinated crop. The chances of the self-pollination is very rare in onion crop. It belongs to Ariaceae family. In Sri Lanka, major growing districts are Jaffna, Vavuniya, Puttalam, Klinochi, Trincomalee, Monaragala, and Ratnamura. This study of flowers includes initiation and anthesis of flowers, dehiscence of stamens, stigma receptivity, and development of fertilized ovary. This knowledge necessary for crop breeding and hybridization program. Shallot and multiplier are the type of the cluster onion. This is my main objective of my research, to investigate the floral behavior of different cultivars and variety with vernalization and non-vernalization condition to select the good parental line to improve the seed sets on hybridization. Next, materials and methodology. This is my study area. I did my research on Regional Agricultural Research and Development Center, Klinochi. The soil type is red jello latosol, annual rainfall 1434 millimeter and temperature 22 to 33 degrees Celsius. These are the materials I have to select. I selected 5 cultivars, TVM1, MICLO1, MH4, TVM6, and Kunduvellara with 2 treatments, T1 vernalization treatment and T2 non-vernalization treatment. I did my research factorial randomized complete block design with 3 replicates. For vernalization purpose, mother bulbs were subjected in cool temperature at 10 degrees Celsius for 13 days. These are the field practices I did: land preparation, captain treated to mother bulbs, field establishment of bulbs, irrigation, manual weeding, and fungicide application. These two pictures show the differences between the 2 treatments. Here we can see under the vernalization treatment, presence of the flowers higher than the non-vernalization treatment. These are the observations I have recorded: time of anthesis, time of anthedehiscence, days for bud formation, days for first flowering, days for 50% flowering, number of umbels per plant, number of flowers in per umbel, umbel width, number of flowering stalk per plant, flower stalk height, and flower stalk width. Then move to the results and discussion. Floral biology. The onion flower is called as the umbel. This is aggregate of many small inflorescences. In anthesis observed morning 11 a.m. to evening 1 p.m. After the anthesis, the anthedehiscence was observed in the evening time. The peak level of the anthedehiscence was observed in the next day of anthesis, morning 9:30 a.m. and evening 5 p.m. The anthesis begins from outer layer goes centrally in succession. Sheath splits of pressure created by growing flowers inside the umbel. In onion plants, there are 6 anthers present in the 2 walls, the bilocular anthers and superior ovary is present. These slides show the each stages of onion flower from flower bud initiation to up to matured seed matured seed setting. Next, these are the floral images. First one is unopened buds. Second one, unopened and opened buds. Third one, fully opened buds. Then anthers and stigma of the onion flower. This graph shows the number of days for first flowering. The green color indicated the vernalization treatment. Yellow color indicated the non-vernalization treatment. Within 5 cultivars, MHO and MICLO1 show the earliest flowering in both treatments. There were no any significant differences between MHO and MICLO1, and then TVM1 and TVM6 recorded highest days for flowering. In vernalization, onion plants started flowering, at least it takes 17 days for in vernalization condition. But in the non-vernalization condition, it takes on 24 days. From that, we can conclude that the cluster onion was significantly influenced by the vernalization treatment and non-vernalization treatment. This is number of days for 50% flowering. Here, this graph illustrates there were no any significant differences between MHO and MICLO1 in two treatment. Within 5 cultivars, TVM1, TVM6, MHO, MICLO1 reached the 50% flowering. Kundavalara was not because it's non-flowering characteristic. TVM1 and TVM6 also show the not significant differences between them. But these two cultivars are differentiated between from other two cultivars. Then number of umbels per 100 plants. There were significant differences between treatment vernalization and non-vernalization treatment. There were no any significant differences between MHO and MICLO1 germplasm. When compared to 5 cultivars, these two MHO and MICLO1 cultivars exhibited greater flowering ability. These two pictures show the differences of two treatments. In vernalization treatment induces the flowering, so the highest number of flowers we can observe in the first picture than the non-vernalization treatment. Umbel width of selected cultivars. Within 5 cultivars with two treatment, MHO and MICLO1 had the highest mean value of umbel width. Compare the vernalization and non-vernalization onion flower. Number of umbel and width of the umbel also high in vernalization treatment. Therefore, a significant effect on umbel width by vernalization. TVM1 show the high susceptibility for anthracnose flower stalk during my research period. Then move to the number of flowers in the umbel. Here, vernalization significantly influenced the produce more flowers. Variety MICLO1 produces the highest number of the flowers on the umbel. But Kundavalara was not flowering ability observed in Kundavalara was not flowering onion. Then number of flowering stalk per bulb. TVM1 bulbs had the highest response for vernalization. There were significant differences in treatment vernalization and the influence of flower stalk production in onion MICLO1. Then flower stalk width. This graph showed the MICLO1 had the highest flowering stalk width. There were no any significance between the TVM1 and MHO, but these two cultivars significantly differ from other two cultivars. MICLO1 had the greatest significant mean value of width, nearly 8.65 millimeter. Then self-pollination and cross-pollination also I take additionally for my research. For my research, I selected 3 cultivars, TVM6, MHO, and MICLO1. I rejected 2 cultivars, TVM1 and Kundavalara, because Kundavalara is not flowering ability onion, and TVM1 during my research period, it's mostly affected by the anthracnose on flower stalk. That's why I selected TVM6, MHO, and MICLO1. In the vernalization condition, there were no any significant differences between these 3 cultivars with 2 different treatments. The self-pollination chances of the self-pollination also rare in the onion crop. And likewise, the seed setting also quietly low in the self-pollination. Then cross-pollination. Onion plants are mostly cross-pollinated crop. So the honeybees flies actively increases the seed setting percentage. Here, the vernalization treatment significantly influenced on the cross-pollination. Vernalization increases the umbel size, a number of the umbel presence on the umbel. That's why the active flies, honeybees are actively did the pollination. In vernalization condition, the higher seed set percentage was observed, 82.36%. In non-vernalization condition, lower percentage, likewise 75.49%. Finally, I concluded my research. Vernalization was one of the most effective treatment for inducing flowering ability in my study. The higher flowering observed in MH4 and MICL1. MH4 is the multiplier hybrid one. This one improved line and hybrid one with higher flowering ability. TVM6 show that tolerant to major fungal disease, mainly anthracnose and bulb rot. During my research period, the TVM1 was mostly affected by the fungal attack. And the Kunduvellara had the not flowering ability in 2 treatments. Finally, according to my floral study results, show that MH4, MICL1, and TVM6 are the best for the hybridization process. And using these 3 varieties of cultivars can increases the success of the hybrid seed production. Thank you.
Yes, thank you, Aradhana. Now the time for discussion.
Okay, ma'am.
Hello, Aradhana.
Yes, ma'am.
Yeah, can you take the slide you mentioned about the cross-pollination?
Yeah. This one.
Yeah, previous to this one.
Okay.
Yeah. But your finding stated that there is no significant difference, isn't it? Vernalization, non-vernalization, both
Yes.
have the similar cross-pollination ability. Am I correct?
Yeah, vernalization, the small percentage influence on the vernalization, cross-pollination.
But your statistical analysis stated both are similar, no?
Yeah, ma'am.
Yeah.
Because it's onion plants are mostly cross-pollination crop. That's why the percentage also nearly likewise, almost nearly 82 and 75.
Yeah, that's what. But statistical analysis saying that no difference, isn't it? So
Yeah.
that's what we can't say that vernalization induce the
Yeah.
cross-pollination, isn't it? Though that
Yeah, ma'am.
slightly the percentage is higher than the non-vernalization condition, but the statistical analysis saying that there is no significant difference. So that my question is, why have done this experiment? That what is your intention to do the difference between, to find the difference between vernalization and non-vernalization condition?
Yeah, this cross-pollination and self-pollination I did for my additional, this is for further knowledge. In vernalization induces a flowering ability. That's why cross-pollination induces the vernalization treatment. The umbel size flowers also higher than the non-vernalization condition treatment. The number of the flowers present on the umbel also higher than the non-vernalization treatment. Though, so the honeybees active also high. The honeybees actives, the seed set percentage also higher than the non-vernalization treatment. I found that. Okay.
Okay, so to increase the seed sets only, you have
Yeah.
done now.
Yeah,
Other
ma'am.
than that, self-pollination or whatever it is, cross-pollination, both are similar, statistically similar. Am I
Yeah.
correct? Huh?
Yeah, ma'am.
Yeah. Are there any problem from the farmers that seed sets in the onion is few?
Self-pollination not self-pollination not successful.
Yeah.
Cross-pollination is okay, ma'am.
Yeah, that's what I'm
Successful.
asking. Are there
Yeah.
any that complaint from the farmers that seed setting, because that vernalization that induce the seed setting, no, the flowering.
Yeah, ma'am.
So then seeds, you can increase the seeds.
Yes.
So that's what I'm asking. Are there any complaints from the farmers normally when they are without previously before doing this vernalization and non-vernalization condition? If some problems are there in the environment only, we are doing the research, no?
Yeah, ma'am.
That's what I'm asking. If any complaints are there among the farmers that seed settings in the onion is less. Are there any complaints?
No, ma'am.
Okay, it's okay. It's okay. Okay, Ardhana. Fine.
Thank
Aradhana,
Yes, sir. Yes, sir.
in your conclusion, you showed something related to the fungal attack. In the results
Yeah.
and discussion section, have you included those results?
Yes, sir. Under the flower, these graphs, Kundavallara not plus not zero, sir, because I put this zero. Kundavallara was not flowering ability. In this graph, TVM1 show the very low level of number of umbels because it's my research. This TVM1 mostly susceptible to anthracnose. That's why I put it there.
So, but that is not your conclusion, no? So that may be the reason for that result. So it's better to avoid that one in your conclusion, no? That is not
Okay.
your conclusion. That is, yeah. And another one, what is the reason that Kunduvallara shows that much of very poor flowering ability, those type of things?
Usually, Kundavallara was commonly not flowering characteristic onion.
Uh-huh. So how it produces that tubers or that one? It doesn't have that flowering ability. Is that correct? Kunduvallara?
Yeah, the Kundavallara, my research times, Kundavallara also affected by bulb rot also. That's why
Ah.
this not successful. Mostly all plots
So,
are destroyed.
so that is the reason. So that is that didn't produce any that flowers, so the umbel and the final products, no. So
Yeah, that's yes.
that is the reason. Okay then. Okay. Arvind.
Thank you.
Any more questions? Okay, if no more questions, thank you, Aradhana. Now I call upon fourth presenter, Mr. Jayasinger. Please present your presentation. Optimization of artificial growth condition of Fusarium oxysporum and screening the resistant bean germplasm against Fusarium wilt. Please do it.
Thank you, madam.
Yes, Aradhana, can you stop your sharing?
Good afternoon to you all. I am RKPP Jayasinghe from Faculty of Technology, University of Jaffna. This is my research topic, optimization of artificial growth condition of Fusarium oxysporum physiology for screening the resistant bean germplasm against the Fusarium wilt. Common bean are very nutritional and popular vegetable around the world under the family Fabaceae. It is widely cultivated in upcountry intermediate zone, especially in Badulla district. In this zone, the temperature, relative humidity, soil type and climate is more suitable for flowering of beans. There are many diseases such as rust, reduce the yield and quality of bean in Badulla district. Fusarium wilt is one of them. The pathogen of Fusarium wilt is Fusarium oxysporum physiology that primarily found in the soil. There are various ways to control this, such as fungicide usage, cultural practice, fumigation, producing varieties. Germplasm of bean can be used for create Fusarium wilt resistant bean variety by screening method. In my research objectives, to identify the Fusarium resistant bean germplasm for Fusarium wilt disease and the selected of best artificial growth condition for Fusarium oxysporum physiology for inoculate the bean germplasm. As a growth condition, the growing temperature, growing media and growing media pH can optimize. Fusarium wilt is the one of major disease faced by farmers in upcountry, especially in Badulla district. Fusarium oxysporum cause agent of disease that one of soil-borne fungi that block the vascular systems in bean crops. Due to this disease, the yield and quality of bean are reduced and the management cost also high in this region. Not only that, by the using chemical fungicide to soil, The soil fertility also reduced in the land. These are some pictures show the fusarium wilt disease affected field in Uva region. The first one is Badulla, next one in Mahiyangani in Uva region. The third one is Valimada in Uva province. The fourth one shows the Bandaravela field in fusarium wilt in Uva province. The last one is Diyatalava. Justification: growing resistant varieties has been recommended as the most effective, eco-friendly and easy way to control this disease. The screening of bean germplasm for fusarium wilt resistance is the initial step to identify the resistant variety. For this process, Fusarium oxyforum piscioli should artificially grow and inoculation the root system for beans. From the screening of bean germplasm will produce Fusarium wilt resistant bean variety. These are the materials that I use for my research. For inoculation, I selected 6 germplasm such as BWB1, BWB2, BWB3, BWB4, BWB5 and BWB6. Here BWB means the Bandaravela varieties. Location: the experiment was conducted at Regional Agriculture Research and Development Center, Bandaravela from February to May 2022. Experiment was carried in vivo method and in vitro method. There are 20 treatments such as nutrient agar 30 degrees Celsius with 5 pH levels and the nutrient agar in room temperature with 5 pH levels. And the PDB media such as 30 degrees Celsius and room temperature with different pH levels.
Formation was observed in both.
This is the methodology for nutrient agar media preparation, and this is the methodology for potato dextrose broth media preparation. This is the preparation of protein media for bean germplasm cultivation. Here I used to sterilized soil and coir dust because some soil can presence of the spores of fusarium. This is the inoculation of Fusarium oxysporum piscioli spores to plant by using sterilized equipments with a root cutting method. In my research, the data collection, the macro and micro spores of Fusarium oxysporum piscioli were counted after 21 days of culturing, and the foliage symptoms were recorded. These are the foliage symptoms in bean plants. After that, the vascular discoloration of the plant also recorded. The disease stem establishment also done. These are the fusarium spores are growth in the PDA media. Disease reaction was assessed 14 days after inoculation using a severity index based on the foliage symptoms, and inter-examination of vascular discoloration was performed. Macrospores counted data was collected and summarized by Excel packaging, and it was analyzed by SAS software. This is the final results. According to DSI index, the BWB1, BWB4 and BWB5 germplasm reacted as a resistant varieties, and BWB2, BWB3 and BWB6 come susceptible varieties with DSI index and vascular discoloration. For the best media as a Fusarium oxyforum culturing, the T2 treatment, such as which is the nutrient agar under 30 degrees Celsius in pH 5.5 shows highest amount of microspores, even though there is no any significant different among the treatment T2, T1 and T5. Next to that, the treatment T6, which is nutrient agar room temperature in pH 5, shows the better macrospores. treatment 15, which is the PDB media. Room temperature in pH 5 shows the lowest number of macrospores. In my research conclusion from this research confirmed the bean germplasm BWB1, BWB4 and BWB5 are the resistant to bean germ wilt disease. Nutrient agar media under 30 degrees Celsius pH 5.5 is the most suitable for artificial culture of Fusarium oxysporum physioli. Suggestions: the test can be performed using a large number of bean germplasm to minimize the recommendation of farmers to susceptible for Fusarium wilt disease. The disease acceleration for the Fusarium wilt disease should be used for the source of resistance in breeding programs to develop resistant cultivars. I like to wish all to all help me to success my research. Thank you.
Thank you. So now the title is open for discussion.
Ms. Jayasinghe.
Sir?
So you mentioned you included the two different types of temperature, no, room temperature and the 30 degrees Celsius.
Yes,
So
sir.
what was your room temperature during that experiment period?
It is 25 degrees Celsius.
So it's better to include that one, no, in your paper or in the presentation. Then only the audience will understand, no. So
Okay.
that's the temperature you used, okay. And
Okay.
another one, so you used 6 different bean germplasm. So in the results and discussion table, the very first table, I think. So can you open that results and discussion very first table? Okay, so the disease severity index and the vascular discoloration, two parameters you have shown here. But didn't you analyze those data statistically?
Disease severity index I calculated by the using of the observation of foliage symptoms. Here I
Yes.
not mentioned that.
So based on that just observation, you have categorized susceptible, resistant, no?
Yes, sir.
So it's better because you have done the experimental design with the replicates. You have enough data. You better analyze the data statistically and present your results. Okay.
Okay.
And another one, the next slides, please, regarding the media. So here the macrospore amount, the T1 and T2, but both are statistically not different, no? So, but you have mentioned the just based on the value, T2 is the best media. Even the T1 also the best media because between T1 and T2 there was no significant difference.
Yes, sir.
Okay. Okay. You got the point, no?
Yes, sir, I got.
Okay, and some suggestions. I have seen a lot of spelling mistakes in your slides. So just an advice. I'm telling that one. So you have to improve those type of things. Okay. Otherwise, okay, good.
Any more questions? Okay, thank you. As no more questions, session 1. So thank you very much for your cooperation on the presenters. You have done well your job, and for your cooperation also. Thank you very much, panel members. I thank all the panel members for your contribution to have this session very successful. Thank you very much.
Dr. Bhairavan?
Yes, sir. Yes, sir.
Okay.
Okay.
You can start.
Okay.
Dr. Nisansala Vidhana Patirana also will join shortly with you. You can go ahead.
Okay. Okay.
Okay, thank you.
So good afternoon to you all. My voice is audible?
Yes, we can hear.
Yes. So this is this second part of the technical session 1 crop protection technology. as I am a panel member and until the joining of these other panel members and the chairperson, I am going to continue this session. So therefore, in this session, there are 6 papers going to be presented by the different authors. So the first paper of this session 2 is ISA 012 with the title of Analysis of Phytochemical Present in Selected Plants Using Water and Methanol-Based Extraction. This is the authored by S. Karunakaran. Mr. Karunakaran, this is over to you.
Okay, sir. Thank you, sir. Sir, good afternoon to you all. Okay. This is the research findings. The analysis of phytochemicals present in selected plants using methanol-based extractions. I am Mrs. Karunakaran, one of the authors of this paper with Dr. Niranjana Pradney Fernando.
I think she's not there. She has left from the connection. I hope that there may be a network issue. So then we can continue with the second paper. The second paper is going to be presented by the P.A. Shiromia on the title of Relative Effectiveness of Vermivotion Phytophagous Insects of Dodlong Bean, the Vigna unguiculata variety Bushita. Shiromia, this is over to you.